So we couldn't hear you. That's all.
Shared it.
Yeah, I off there for a bit, but I don't know why it's not sharing. Is it sharing?
Because we could see the people on Zoom.
Yeah. Okay. Yeah.
Yeah, the screen has been.
Thanks for confirming.
Oh,
Okay,
thank
you're
you so
saying-
much for confirming that the
Thanks.
screen had already been shared. If the audio had been cut off, what we went through, what we went through were basically what was in the slide. We just gave an overview of what are the brain cell atlasing efforts that's currently underway, what has been built up in the field, what are the disease atlases, brain cell atlases available. Awesome. Okay. Moving into this question, we were in this. Let me see if I can make it bigger. If I switch to slideshow, are you still able to see the slides? People in the Zoom? Yes. Okay, great. Okay, so has any, yes.
Would there be some ethical concerns if we start including diversity in brain samples? Would the data be used to correlate intelligence and diversity?
So the modality is that, yes, very valid question. If you go on to do some electrophysiological analysis or add other modalities, right? But the modality is that we are currently, or at least the ICCN and HCA is currently looking at single cell spatial, HCA maybe. Not too sure if we would, if that's enough to put together to not to look at intelligence or the correlation with other factors. Not that it's not possible, but I think there are ethical committees within each of these consortium efforts in place. There is a framework there that does not allow people, or does not allow science PIs to.
Divergence. I think people might be more accepting if you think about it in from that angle as opposed to intelligence or consciousness. Yeah.
Neurotypical samples. There is no such thing as a neurotypical in the entire world, but again, healthy samples, right? So, and that within that healthy distribution, there is different neurodivergences included as well.
Actually, I'm kind of still on the very first axis, which is genetic diversity. Like I'm stuck thinking about it because in my head I keep trying to think about any brain disorder that actually shows differences, on this axis, and I wasn't quite sure. I don't have an answer in my head, so I was wondering if anyone actually know of such conditions. Yeah.
It just reminds me of okay. So I've been in different places. I really wonder why is the Philippines very poor and has been left by a lot of Asian countries in terms of economy. And one thing I suspect is about the effect of colonialism on the mentality of the Filipinos. That's one hypothesis. Okay, so maybe that's something that could be answered in the future. I don't know how many years we would wait for that to see that a pattern in the brains of Filipinos who were, yeah, that's it. So that's one thing I suspect. And yeah, because I keep saying I haven't seen any countries in the world which has more potential than the Philippines in terms of resources. But why? Aside from corruption, of course.
Yeah, to build up for genetic diversity and I think you're kind of bridging genetic diversity and environmental factors, which is great. Let me bring back the conversation to genetic diversity. First, that talking about Philippines, right? Let's take it from there. So there's X-linked dystonia Parkinson's, which comes specifically, which is found only in one particular island in the Philippines. So that's a genetic disorder, and that's a brain disorder. People are putting together. People are using the Wang represent as the HCA represent to study what cell type proportions change in X-linked dystonia Parkinson's. Yeah. What other you were asking about what other diseases are would benefit from having this genetic diversity while building the atlases? Here's a few examples. Like for example, this one, right? In Parkinson's, the GAB variant, there's a very strong bias between the African and the American cohorts, and that's why exactly we need to include. We need to have a good geographical representation. It used to be diversity, now it's geographical representation within the sample.
Thanks for reminding me. I'm clearly sleep deprived.
Okay. Access to, yes.
Sorry, I was just going to mention for the neuropsychiatric disorders, which are super complex genetically, pretty much everything we have currently is European risk, and it's going to, I'm sure, look quite different around different places around the world. So that's one clear reason.
I was just going to clarify on that a little bit for people who don't think in genetics terms, right? So we know that polygenic risk scores that have been built in European populations, I think this is what you were already talking about, which is why I hesitated to take the mic again, but just don't do well in non-European populations, right? So there's different combinations of variants probably that are then contributing to it. And so being able to parse that out and look at that. And I think part of it also may not be disease driving, right? But having a clear representation of the diversity will allow us then to be more representative and provide effective clinical care for, I think, a broader range of people. So I think that's the direction that I'm coming at the diversity from. So I thought I'd just add that in there before we move on.
They have more.
Questions of people.
Yeah.
Do you access to banks and where do you get your samples from? The way your organoids came from a commercial source, right? The cell line stuff. How about in the government?
I'm at the Garvin, but I don't use brain banks very much. So a lot of the work that I'm now funded to do, but haven't started yet, is going to be iPSC derived bipolar disorder work. So I'll be generating 50 new iPSC lines for that. Mike and I also have some collaborators at the University of Melbourne who have over 100 lines across neuropsychiatric diseases, specifically phenotyping for psychosis, and then some other collaborators that have also worked in that space. So across Australia, we definitely have some neuropsychiatric iPSC derived lines. I might look at Mike. Are there other ones that I'm maybe missing?
I don't think there'll be a large number of lines currently, but probably there's potential to make many. And then we've got reasonable brain banks in Australia. I think especially schizophrenia, there's quite a lot in New South Wales, near Sydney. And then where I'm in Melbourne, we also have brain banks, which again has a reasonable number of neuropsychiatric and neurological ones. What we actually lack are controls.
Because Vican and Mental-FS version 1 samples are primarily dominated by neurotypical or healthy brain. So yeah, and the data is really up for public access. So coincidentally, I've been walked in.
Yeah.
So please contact Ed or one of us if you want to get access to Adam Brain Atlas data set or any of the Vican, PICCM Mouse Atlas or the Vican Brain Atlas data sets. They are great resources.
So Yukinori left a very interesting caption on his acknowledgement page, and I checked it out. And apparently Japan has a very large brain bank. Yeah. And yeah, it's kind of missing from the list. So I was wondering whether it would be, interesting to explore the option.
They do share samples.
Yeah, exactly. Yeah.
Of the data set. I do know that the Neuro Biobank, the 6 Neuro Biobank across US, the data is on NEMO. Isn't that it's NEMO and also the Biobank data center?
Yeah, the data itself is in Nemo.
The data itself is.
This is points.
So the BICCN data is available on NEMO for everyone to download. The BICAN data set, probably some of them are there. And then very soon, in a couple of months, keep watching for updates. You'll get a new wave of data sets there, primarily from human and a couple of other non-primates as well. Yeah. How about yes?
Do we have any idea on the impact of the sampling that has already happened as the biobanks were formed versus how much that could kind of like paint a picture of variation that is existing just because of how the samples were acquired? And if yes, would it be helpful to create like a mitigation strategy such that those samples could still be used, but that somehow the bias would be acknowledged upfront?
How are we the technical variation or other non-biological variation that focus on biological variation? I do know that the human selectors for Xiaoqun as well as the BICAN has put together a structural framework for computational pipelines, and they also provide CCF, like common coordinate frameworks. The data comes in CCF formats that others use where sources of variation are both. And
Yes.
Yeah. I mean, I think there are a number of challenges to this. You know, some of them are relate to how the tissues themselves are prepared. Some of them have to do with anatomy. And there may be other technical factors associated as well that make it sometimes challenging to compare. Maybe I need to have it closer, that's all. So actually the Brain Initiative Cell Atlas Network that I was describing tried to take a different strategy towards that, and it could only really do it because it had the consortium behind it and the National Institutes of Health kind of forcing it. They recognized that they needed to have consistent samples for all of the projects that were going to be funded. So they contracted with the Neuro Biobank. That's sort of a federation of brain banks across the country. And actually the idea there was no geographic location in the US can actually capture population demographics, but collectively they could. But they were also all preparing their brains in different ways and not collecting metadata in similar ways. So we spent a solid year at the beginning of the consortium to try to bring the best in practice methods so that they could be prepared in similar ways across the banks, that we would capture common data elements across all of the different banks. And then those could be distributed across the various projects. And also to set up a whole infrastructure for standardized sequencing and processing of that data, and for people to have to document where their samples came from, photo document it, map it into a common coordinate framework from the beginning in order for it to enter the system. So it's an elaborate system which works well, but is not yet extensible beyond the Cell Atlas network. So I think an interesting thing to consider is which of those things could actually be democratized in a broader way. For example, even just the brain preparation, there are much improved ways for doing brain preparation than are common in most places. And they result in higher RNA quality, higher tissue integrity, better ability to do spatial analyses and things like that. So that's like really worth the investment up front, because there are a lot of samples that are just, you know, you're going to have problems because they were prepared differently or they weren't stored properly or whatever. So I think brain is unusually difficult in that respect.
Point about brain banks. What about in Asia? Are there brain banks I know Singapore is building a brain bank, and do you know how many percent of
Okay. Yeah, I actually I know in China, 10 years ago there are not very much brain bank, but right now, I think they have a kind of brain initiative. Give pretty much a funding to support collect the brain tissue in China. As I remember, there are a ton of donation line up. Like people right now is willing to donate. They line up. Pretty much right now they collect all 100 or 500. Yeah, so it's a ton of people line up. It's a complete change of the mind compared to 10 years ago. Huge, I would say.
As a community, as a neuroscience community, I think we need to do a better job of reach a perspective sample collection.
But as I think Ed talked about, because pretty much we might need a sort of like a protocol, how to standardize, how to deal with the process. It's quite the version going to be work. How to section, bring into different part.
We did find out in some preliminary analysis that we did with close to 200 brains, we did find out that the BMI, the postmortem interval, did not have an effect on the RNA quality. However, the brain pH did. So that was one good way to screen samples that you want to keep aside for spatial or other in-depth genomics analysis. Yeah. I think BICAN shares those resources freely with anyone who requests them. Yeah. Okay. So before we move on, I just wanted to also touch upon the challenges in case people are using the brain banks for. Have you ever tried getting access to brain tissues from another brain bank, like within Australia? Or have you gotten access to prepared nuclei? Are people willing? I know cross-country is a problem, at least within the country. Have you tried such initiatives between Melbourne? No. So it's strictly restricted to a particular institute or a university. There's no sharing of samples. It's just not the area that we've worked in yet. Okay. Okay.
I think some country can share. Also, time with the policies.
Yes. Yeah.
Remember, United States can share in them, so they can. Now we're there actually. They can transport the tissue cross country. But for us, for Asian problem.
For Asia, yeah.
I think it's
strictly not allowed. Yeah. Take away.
Okay. I think we bring up the key discussion question. The second question is like how the immediate priority, which neurodevelopmental and disease method should be priority for population specific, like focus area, where population specific origin is relevant, and how can we cross species cooperation be leveraged, like use different animal. Yeah, it's free to discuss. I mean, for what I know, right now, China, they have a initiative first, but right now started focus on autism disease. Autism disease, a lot of. Another second is especially in Asian right now, it's different. I think the aged population will be a very serious question, especially in the brain. number going to be accumulated more and population. Asian right now.
Clarify your question. I wonder if you could clarify your question. I mean, any of these diseases is very difficult to take on. So is your question societally, which are the most important, or for HCA, which would be the most important or the most relevant?
Not only for the HCA, right? For people, pay attention, right? For society, for.
I guess you do have an important point, like how are we going to prioritize what's good at large, but also what with HCA is focusing on prioritizing.
But I mean, let me give you an example. Like Alzheimer's disease, of course, is a super important one for aging societies. But there's a huge amount of funded research already in that area with consortia upon consortia working in those spaces. And so they have the advantage of access to many tissues. But they also have the disadvantage maybe that they already have established communities that are doing a space for HCA to make an impact.
I think we did have a table up there that talked about, the SEAD and size effort and Manolis-Kellis's effort. And there's considerable amount of work being done on this. So what would be our focus? Does anyone in the room have a particular disease or neuropsychiatric disorder that you want to focus on?
If I could just make one other point, sorry. At least in the US, we have very limited access to developmental tissues. Hard to get those. And in some other countries, it's a lot easier to get access to both prenatal and postnatal development. That might actually be a place where a global approach to bring together samples can be really effective in a way that I don't think can be done.
That's pretty much a great idea because it's different countries have different,
Yes.
Are there quite a few same?
I don't think so. And Julia, your samples are in your department, right? Do they include the other samples, like at least 5 to 10?
Yes,
Five
so,
to eight.
so in my case, I'm working with the active speech, right?
All right.
So they're not primary tissue. But when I first thought off the top of my head is not so, right? But like my head, do you know that specific communities and are already heavily invested this effort for occupancy. So I thought you raised a really great point, which is how each contribute and in what
globally.
ways. So I really hope that certain countries are more, they have better access, early development tissue and
Do you see any issues with sharing, with data sharing, by those countries that need such efforts? Would there be any hurdles to data sharing, or would that be easy for the international?
I think most of the challenges since
All that stuff. I can't speak for European countries.
How about in Australia? I could just ask about how we see this to share project to project, but I think that your group sharing probably we've shared open access to medical work, but we've also been down on that to make controlled access so that regulation around this thing. Do you want me to go on?
About the cross species. Primate,
I can answer that. I was just going to say, I think Ed's point before, I'm just going to sort of third that. I think that's a really good one because, you know, Alzheimer's, for example, big consortia. Then there's maybe a question about some of the neurodegenerative stuff. If you've got post-mortem end state, is that really the best thing to make a big atlas from? Really what you want to be looking at is 30 years earlier, and you don't necessarily have that. So neuropsychiatric is really good, but again, there's PGC and so on that, you know, they're doing a lot of that work. But so neurodevelopment's really good. No, I think cross species is quite good as well. So we do a little bit of stuff on marmosets, for example. So that obviously is often a bit easier to get hold of because the ethical concerns are not so great.
White matter. Any interest in white matter here? White matter? Okay. Okay. I think what Xiaoqun means is where genetic
Diversity, yeah.
fine or is that what-
Yes, it's genetic background diverse.
Well, I think the other thing that I would maybe add to this discussion is it would be really interesting to focus on diseases where we do see differences in frequencies in different populations, and I feel like that might be a good way to focus on maybe a disease to see what's that contributor. Is it environmental or is it a genetic contributor? And it also might allow us to then focus on diseases that are of interest to those communities and of interest to specific world and get more of that diversity in that way. So I thought that might be another good way to choose areas that we could focus on.
The absence of variation that we could be interested in, right? Like genetic diversity, environmental, so clinical subfield, like drug responder, non-responder, or any other. Right? Does this cover or were you intending to add one more layer?
I guess the other two layers that I kind of think of but probably don't work in very much are age-related differences, which I think we've also talked about with developmental, and then also like urban versus rural. And I don't know how much that would contribute to variation as well. And I think will be a little bit harder. So I think those three that you started off with are probably really good starting points to understand each of those contributing factors.
Crossed over the point where we were talking about the axis of variation and age and sample collection, gender, right? Most of the brain samples that been worked with or I've heard present on is heavily male biased. So is there any, especially for a particular age range, like 30 plus, 30 and above, does anyone in the room have ideas as to how they or some study has kind of balanced from that gender balance this sample collection? Yeah. So, or I scan.
Yeah, I have a lot.
I mean, it's difficult to balance on that because into the medical examiners die younger. In Neurobiobank, if you look at the all demographics, younger ages, it's incredibly skewed. Finally, when you get into the very older ages, versus a bit, then you've got all sorts of generative things. Depends if you want to actually study that as the disease. Much harder. Judgment area you need. World. A lot to choose from, and then I don't have another.
Yeah, contribute from all different results. Go ahead. Okay. Beyond Atlas effort, how can we translate Atlas discovery into a therapy that benefit all population? Of like how to correlate with a therapist moving from Atlas Insight to biomarker targeted therapy, clinical trial. How can we Atlas contribute to this? I think that's probably the final aim for the open question, especially for the biomarker. I think the molecular marker, a cell type might correlate with for brain, correlate with
While it takes a break, maybe I can quickly share something. One success story I can think of is this Huntington disease, right? So where some of the single cell efforts led to identifying expansion, repeat expansion in one of the spiny neurons, and then ASOs are in clinical trials, antisense oligonucleotides are in clinical trials, and that came out of a brain cell atlasing efforts. Can anyone think of any other?
Yeah, I guess the atlas helps with like targeting. There's just new set of AAV therapies, right? A whole bunch of new AAV therapies, and knowing which cell type to target will help you. And the atlas is the perfect thing to do, like because you can hopefully define your molecular markers, you can define your molecular cell surface markers, and then you have something that you can understand and something hopefully that will be more specific, because that's, I guess, the holy grail, since we are always talking about trying to develop more specific therapies rather than just flooding it with, say, a reuptake inhibitor. So broad, it just damages everything in all sorts of ways that we don't expect.
I think especially for recently there are more and more promising news like for Parkinson's disease, there are cell therapy like iPS derived and all stem cell derived open neuron put into the brain Parkinson's disease. So we pretty much have to know the standard candidate cell. That will probably help a lot. What kind of marker? But right now, I think there are no standard, right? Just a clinical trial, and from Japan, from Merson category. Yeah.
Am I summarizing this right? So HCA's immediate attention is just going to be building reference atlases with the hope that help us in future evolve or help detect out other disorders in psychiatric studies. Is that going to be our immediate, or is HCA the nervous network, the nervous system network, looking into picking one particular disorder or a bunch and then letting go within this network?
I don't think it's happened at all yet. So matter of some discussion of what makes sense here. It's really difficult to get to either biomarker. I think we've got the tools now to be able to understand disease at a much finer level of granularity. But you really need to design sort of cohort studies, progression of that. And you really want to get to biomarkers, ideally, longitudinally collected biofluids from those individuals that you eventually train from, and then you go back and test whether you could. At the moment, those things, it doesn't really exist.
Yes.
You have people just doing postmortem studies, you have people doing living patient fluid studies, and they're not connected. And so it's like a huge problem in the field right now. So it's a little hard to conceptualize of how the HCA could do somehow better on this. Different diseases are really different. Example of neurodegenerative diseases, pathology. Pathology is a quantitative thing that you can measure and use it to have disease trajectory, cellular phenotypes on. Developmental diseases, you don't have that. Right? So now you have genetics, but you never get enough brains for any particular study. But it's not a trivial thing to get to find. Relatively low hanging, find the vulnerable cell line. You have that. That's important. Now people can focus on that.
Like epilepsy, like
Or all the degenerative disorders. And that might lead you to a biomarker, it might lead you to a pathway, it might lead you to help that target. So you have the target, you don't know to.
so the dream to get a biomarker. I would see.
I mean, I'd love to hear some other thoughts on this. How could we accelerate by working together?
Yes.
Rather what psychiatric disorder focused on. I'm worried, but I would say for working in neuropsychiatry, I'm probably.
Yeah, I would say Garvin's not well known for working in neuropsychiatry. The places that are more well known for it across Australia, I would say, are Laurie. I'm looking at Mike now to make sure that I'm not saying anything wrong. University of Melbourne, that's where a lot of our collaborators are. And then a lot of neuropsychiatric genetics in the University of Queensland in Brisbane. But I will be focusing on bipolar disorder, which I partially selected to start looking at because it has a really high genetic heritability, which allows me to look a little bit closer at the genetic interaction with pathways. Previous work for me has been in major depressive disorder, which is only the 20% heritable, which makes it a little bit more challenging. So I've pivoted a little bit there. Yeah, does that help?
Just want to take a stop of, what's the interest in the books that we've been documenting all the conversations and helpful to put up an action item and follow up on people who are interested, who showed interest. I'm told we only have a minute left.
So the time is coming.
So if you have any thoughts of how we could, fill in any
Technical
gaps,
challenging, right?
in our current tools and workflows, feel free to write to us. There is, I hope the Neuro network within HCA, the email is still active. So please, you can write to that email address, or you could write to us, and then we pass along the questions. And with that, I don't think we have time for this. So with that, thanks everyone for your contribution, and look forward to sending you some of the action items from this. And probably it's going to be an ongoing conversation. At the Neuro meetings, the HCA Neuro meetings, does it occur among the friends or?
No, I have to say that the Neuro network is more or less synonymous with the NIH Brain Initiative. So if people are interested in participating, let me know. There hasn't been critical mass to really start such meetings outside of that. It's also possible for people to become honorary or sort of members of BICAN and participate in that as an alternative. So, but if there's critical mass, we can actually start to have meetings.
Very nice. Yes, yes.
To build this community. Thank you, everyone.
Thank you, everyone. Yeah.